Welcome back to our discussion, Evolving Biosurveillance in the Post-Pandemic Era, sponsored by Nobles here on Federal News Network. We're speaking with Catherine Jennings. She's the lead scientist at the Applied Science Research Center and senior manager for the life sciences team at Nobles. I'm Tom Temin. And Dr. Jennings, beforehand, we were talking about the couple of studies that were done with respect to the polymerase chain reaction work and also the genome sequencing of COVID vaccines. And I want to ask you about that latter one, the vaccines, and what you have been able to study and learn about the efficacy and effectiveness trends in gender, in geography, and other aspects of the population that's receiving these. That sounds like really crucial information for federal planners.
Apologies, Tom. It's not so much that we've been sequencing the vaccines, but we sequenced a population of humans that had been vaccinated and had infection anyway.
Got it. Okay. Well, good.
So what happened, we were receiving whole genome sequence from our partner laboratory, and then we were doing the bioinformatics analysis. And we found a cohort of a little over 30 patients who were what we call vaccine breakthrough cases. They've been vaccinated, we think they had been boosted, but they still came down with infection. And when we looked at those whole genome sequences at the base by base level, we were able to determine that this cohort had a constellation of mutations that hadn't been seen before. And we think that was probably contributing to the breakthrough. The functional studies haven't been done, so we can't say that for sure, but we found specific changes in 3 different places within their genomes. And what I thought it did was highlight some areas for future study to better understand the biology, both the virus and the changes, and how vaccination may be putting pressure on the virus in these populations and contributing to changes over time.
And is that something unique in the history of all of this? Because when you think about the vaccines that have been developed in the past 50 or 60 years, in measles and polio and so forth, those have been largely eradicated. We don't get the mass numbers of breakthrough cases that we've seen in COVID. I'm one of them, and live to tell the story. But that seems to be something that is emerging with this particular pandemic. Or am I wrong? Or has this happened before?
It's happened before, but I think a better comparison and framework for us to think about is influenza viruses. So they're similar in that they're both RNA viruses, and influenza virus changes very frequently. And so that's the reason that we have to get a seasonal vaccine every year, is because the mutations it sustains. It sort of looks like SARS-CoV-2, maybe similar in that it just mutates fast enough that we will require boosters with some regularity, as opposed to other viruses that are maybe larger double-stranded DNA viruses. And because of that molecular biology, they change more slowly. So we can get a booster every 10 years instead, or possibly never, and still be protected.
But no chance right now of all of these different vaccines mixed into one big dose that you get and you're good to go?
I don't think so, not just yet.
All right. So from the learnings in studying the DNA of the people that had breakthrough infections after vaccination for COVID-19, what can we take away as far as what the new progress or new process might be in biosurveillance? How does that translate back to government agencies tracking this kind of thing?
I think it speaks to the ongoing need for us to continue surveillance. Even on SARS-CoV-2, we have more than 1.6 million whole genome sequences. Because it changes all the time, though, we need to keep tracking it. And I think it's really kind of highlighted the need to do whole genome sequencing at scale for many different infectious agents, partly because it helps with building those PCR-based detection assays that stay current, but it also gives us tremendous insight into the genome of these pathogens so we can develop more effective vaccines and medical countermeasures.
Up until now, and especially this came very clear in the COVID-19 crisis, the development of vaccines, at least from the layman's standpoint, appears to be something episodic. There's an outbreak, there's a big national drive, And we were lucky to be able to develop this particular COVID-19 vaccine relatively quickly. Some vaccines have taken 50 years to develop after identification of the virus. So does this then, as we're talking about the evolution of biosurveillance, it seems like this should become a more continuous activity, whether there's an outbreak or not. Is that where we're going here?
I think that is true. One of the remarkable things that worked really well in the pandemic was the development of these mRNA vaccines, that happened so quickly. So it was a platform technology that had been in development for years, and they applied it to SARS-CoV-2 and it worked out really well. The other thing that happened that was unique in this pandemic compared to, say, the vaccines that took decades to produce is that a lot of those clinical trials happened simultaneously. Ordinarily, they happen back to back. You do a study for years, and at the end of that, you start another kind of study. There's a lot of risk with doing them simultaneously, but in an emergency, we had to do it that way. So what I'm curious to see is how the pharmaceutical companies and the government decides to manage that risk, and how willing they are to do those things in parallel going forward, even in the absence of an emergency, to try to increase the throughput and flow through and development of vaccines that we currently just don't have.
And putting on your Cyberni practitioner hat here in the bio area, what is the infrastructure, do you feel, in the field? Is it adequate for doing this kind of continuous, almost like cybersecurity, continuous diagnostics and mitigation type of process in the biosphere relative to the episodic way we do it now? And all of a sudden there's trillions of dollars available. But maybe if we had some kind of a continuous program, we would know things way more in advance, as we discussed earlier.
I think what you're describing is symptomatic of our public health infrastructure at large. We really need a more robust infrastructure. We need coordination at a grander scale. We need continuous funding at a greater level, both for the vaccine and medical countermeasure development that you were just asking about, but also for the detection and maintenance of public health, because it had just kind of not been funded at the level. It had been neglected essentially for the last 50 years. And I think the pandemic has really highlighted that while we have policies and we have guidance documents in place, we've sort of repeatedly failed to learn the lessons from the past emergencies and started really investing in those ongoing activities that would lead to public health and some prevention that would save a lot of money as opposed to being reactionary.
Right. So a beefed up infrastructure then would make future outbreaks something easier to deal with and less expensive to deal with, because a baseline, if you will, of knowledge and history will have already been built thanks to that infrastructure.
Exactly.
And one of the trends in other domains of medicine is tailoring it for people that have felt underserved or have not gotten the treatments that are germane to what their situation might be. For example, treatment of women might be different from men for certain diseases. Different racial groups, we're finding that maybe medicine and its application has to be tailored. Are we finding this maybe for vaccines and these virus-related types of illnesses? Because you mentioned that there were some trends in gender and geography with different variants, and it's a more of a kaleidoscope type of picture we're getting.
Right. I think that is another really interesting area that has started to grow out of the pandemic, and that is understanding that what the human host brings to this battle with the infectious agent is almost as important as what is the infectious agent and what is the response, what's the outcome of that infection. And I think it's really opened doors to a lot of new science that can be done, and I think it will be done. And that's one of the important things about whole genome sequencing is we can also do that of humans to better understand what is the diversity of our genomic backgrounds, and how does that affect both the infection, but also treatment and vaccination. It will almost certainly matter what our genetic backgrounds are and what those outcomes ultimately are. And I think it will lead to eventually better vaccines and better medical countermeasures.
And somehow this will all have to translate down to the individual patient and the doctor, because if we're going to have mass types of sampling and sequencing consistent with privacy and all of that, what is the methodology here? I mean, when you do the vaccines and the efficacy sequencing and how well it was working, is it from blood samples? Can people spit in a jar? I mean, how do you actually do this in a practical sense to get this large data set?
I think it is all those things. Certainly in the medical community, there's always an interest in non-invasive sample collection. We certainly don't want to put people through any more stress than they already are. And at DARPA and others are funding big studies to try to understand what kind of samples can we collect non-invasively and look at these interactions and understand what those outcomes might be in a predictive way instead of having to wait for them to happen.
And just finally, before we close, maybe just go a little bit more into the idea of linking biotechnology with information technology, because it seems like that's the noblest strength here, is to take the big data aspect of it and apply that in a way that information technology can be used to boost what we know about biotechnology in a fast way that affects more people quickly.
Yes, absolutely. So we do have high performance computing. We have extraordinary talent in terms of coding algorithms and doing data, both the data analysis, data cleaning, building the tools to work with all of this data. And it's one of the ways that we're trying to push the envelope and really innovate the tools that people will need to handle these large data sets and find those actionable insights and take, we call it lab to mission, take these prototype tools and really push them out for application in the field, in the medical communities to be practical solutions to these really hard problems.
And a lot of federal agencies, of course, by law and also by good practice, are starting to retain large numbers of data scientists and data analysts and data officers. Sounds like that should be something that occurs also in the private sector, so that the two sides can really work together better as the next outbreak occurs.
I agree. I had an opportunity to talk to students from the University of Georgia earlier this year. And I told them, I said, if I had to do it all over again, I would learn to code. I would be a much more data intensive biologist than the microbiologist that I am today, because that's where the field is moving. And that's what we're looking for as we bring in young talent.
Well, fascinating discussion, and it sounds like it's going to have some real important, real world practical applications, and we thank you for it. My guest today has been Catherine Jennings. She's the lead scientist at the Applied Science Research Center and senior manager for the life sciences team at Noblis. I'm Tom Temin, and you're listening to Federal News Network. For more information, please visit federalnewsnetwork.com and search Noblis.